So in this lecture, we're going to talk about memory. Now, memory is oftentimes discussed in great detail in a class like cognitive psychology. So we're only going to deal with it a little bit, sort of introduce these kinds of concepts and talk about them in the context of animal behavior and learning. First, there's what's called the multi-store model of memory, and this was first developed in 1968 by Atkinsons and Shiffrin. And it's the idea that we have basically three types of memory. We have sensory memory, which is really quick, just about a second. Short-term memory that lasts for maybe several seconds, but has sort of a limited capacity. Can't remember a lot. And then long-term memory, sometimes called reference memory, and that has unlimited capacity, and we keep that for a long time. So think of sensory memory as maybe like a flash of light that happens and goes away. And you kind of keep that flash of light in your senses for just a little time, less than a second. So I kind of think of these kinds of firework sparklers, and we circle them around in the dark. You can kind of see a circle, but it doesn't last very long. And sensory memory, unless it's relevant, unless it's important to you, it goes away very quickly. It's very fleeting. Short-term memory lasts for a little bit longer, maybe up to 20 seconds, or if it's rehearsed, in other words, it's thought about consistently, or if there's a lot of attention to it, then it might last just a little bit longer. It tends to have a capacity of about 7 objects, 7 things, 7 items. Sometimes we call them a chunk of information. We'll get into chunking here in just a second. Working memory is a type of short-term memory, and some people differentiate them because working memory is also short-term, but it's needed to solve a problem or complete a task or work something out. So if I gave you the letters, let's say B, J, X, and Z, and said put them in reverse order in terms of the alphabet, you'd have to kind of think about it and work it out, keeping those 4 letters in memory, but also switching their order. So we're going to play a few games here with memory. And so what I want you to do is I want you to remember these pictures. Do your best because I'm going to ask you to recall them. Here we go. Okay, now I want you to stop the video and write down all the pictures that you saw. Okay, so if you looked at your list, you probably did something called chunking. So in those lists of images, they tended to have organization characteristics. There were famous people, there were vegetables, and there were animals. And so in your list, you likely didn't just put them in the order you saw them. You probably had George Clooney and Brad Pitt and Tom Cruise, and those were in one part of your list. And then you had onions and broccoli and vegetables in another. And then you had the different types of animals that you saw. This is an attempt that we do naturally to organize content to help in our memory. So dog, tiger, and elephant were placed under a category, a chunk that you said, okay, these are all animals and this is how I'm going to organize it. So long-term memory, well, it's relatively permanent. It has an unlimited capacity. And what we do is we convert through a process called consolidation. We convert short-term memory to long-term memory. And we do that through rehearsal, through coming back to it over and over, to ascribing emotional characteristics, and also organizing them semantically, which we'll get to in a moment. We do what's called semantic coding, which is storing by meaning, and we'll see that in a second. We have mental representation, visual coding. Now, sometimes these are declarative. We've talked about this before. Or explicit memories, things we bring to a conscious state. Episodic memory, these are memories of events that happen in time and space and at a place in our past. We think about it, we can recognize it. Semantic memories are memories of facts and knowledge. And there's also what's called spatial memories, which are memories of things that we remember relative to ourselves. So if you think for a moment, how would you go from where you are now to a grocery store? You can think about it. You can map it out. We make what are called cognitive maps. These all can be declarative because you're bringing them to a conscious level. Then we have procedural or non-declarative memories. These can last a very long time. This is what this class has mostly been about. Implicit memories. These are unconscious. So a procedural memory might be something like driving a car, riding a bike, texting on your phone. You don't think about it. We can just do it as a muscle activity. Non-declarative memories can be classical conditioning, instrumental conditioning, and such. There's a lot of ways of thinking about episodic memory, but one is kind of interesting because it's the way we view or remember or bring to consciousness that memory. So what I'd like you to do is try to remember a trip you took in the past couple weeks. Maybe it was a long trip. Maybe you went to another town. Maybe you went on a trip camping somewhere, or maybe it's just a trip to the store. Think of the place you visited. Think of that memory specifically. Now try to remember something that happened to you when you were a small child, maybe in kindergarten or first grade. What are some of your first memories? How you view those memories can be very different. So the more recent the memory you took a couple weeks ago, you take a field approach to that memory, meaning you see what's happening as if you were there looking through your own eyes. However, if the memory is very old, the episode is very old, we tend to act as actors within our memory. You don't see what's happening in first grade through your little first grade eyes. You see yourself. You're like an outside observer of a small child who is you doing that memory. So episodic memories can shift in our conscious awareness of those memories depending on how long they've been around. Okay, let's try something else. This is dealing with what's called a semantic network model. And we think of these in terms of remembering semantic memories, which are memories of facts. So I'd like to introduce you to this little creature here. It's called a daegu. Have you ever heard of a daegu before? Maybe not. Most people haven't. But now you have. You've learned something new, a new piece of fact. And the little daegu is a South American mouse-like creature. And so now you've learned this new thing. And maybe you want to keep it for long-term memory. So you rehearse it and you think about it and you look at the little creature. But also in creating this new memory, you're going to organize it within what's called a semantic network. So that network might look something like this. So here's a semantic network models. Each of these are kind of nodes. We call them nodes, and we can see how they are interconnected in our memory. So you might have a node for fur, and that has a lot of different information in it, but we might also think of fur and cats and dogs and all connected. And mammals is a very broad node, and it's connected to many things. So mammals go to dogs and cats and rodents. Now here's the daegu, right? You just try to remember the daegu, so you put it into the semantic model. the semantic network. So it looked like a lot like a mouse. So you're going to connect it to rodents, and maybe it was a little bit furrier like a chinchilla. In other words, these are the theories of how we organize memories. You've probably experienced this when you're trying to remember something. Let's say months from now, somebody says, hey, have you ever heard of a daegu? You might think, oh, I'm not sure what that is. But if you start thinking about mammals and then rodents and rats, it's going to eventually connect, and that memory is going to come out. Sometimes we can remember things by thinking about other things that are similar to it. So let's try a little task. I want you to remember these words. Ready? Go. Okay, now stop the video for a moment and write down the list of the words you remember. All right, now you take a look at your list. Which of those did you remember? Now you probably saw the semantic node model that I was creating. These were all things found in a kitchen. Many of you wrote the word fork. I've done this before, and a number of students write the word fork. Fork wasn't in the list, but it was part of the network of things that you think about in a kitchen. And because you hit all the other things in your memory, like a cup and spoon and refrigerator, it also activated that node of a fork. So that was a mistake, and that was an evidence. But let's also take a look at your list. So I've done this before in classes that are in person, many years, and I asked people which ones they remember. And they tend to remember spoon. Which one of these words did you remember? They tend to remember words at the beginning and at the end of a list, and have a little problem with words towards the middle. You can see there's no fork there. This is known as the serial position effect in short-term memory. We remember the beginning of a list and the end of a list. This is also true for many other aspects of our memory. For example, can you name the first president of the United States, or the second, or the third? Many people can, but not a lot of people remember the 21st president. We tend to remember the last few presidents. We remember the beginning and the end of a list. Let's look at other ways that we investigate memory, especially short-term memory in animal species. One is called delayed matching to sample. This is a procedure for looking at memory because it doesn't take into account a built-up of associative strength of a stimulus. It only requires short-term memory to solve this problem. So a sample is presented, and then it goes away. There's a retention interval, we call it. There's some time that goes by. And then two other images are shown. One is the original sample, and one is something new. And that was pretty easy. You probably remembered it was the red one. And this could be done with pigeons, right? Pigeon, the red one comes up, and then it goes away, and then the red and the blue. And in order to get food, they have to peck the red. They peck the blue, they don't get anything. Again, after a lot of training, they get kind of good at it. But the next trial might be something like this. Blue one comes up. It goes away for a retention interval. And then the blue one has to be pecked by the bird in order to get food. Both the red and the blue have been associated with food trial after trial. Red one comes up, red goes away, and then they have to peck on the red key. So neither one of them has a stronger association, but the animal has to retain that information in the short term to get the reward. There's also delayed non-matching to sample, which is a little bit more difficult. Red light comes on, goes away for a retention interval, and then in order to get food, the animal has to remember which one it was, and then also remember the rule, pick the other one, delayed non-matching to sample. There's also different versions of this. Is what's called symbolic delayed matching to sample. If you see the red light, you have to peck the vertical line, but if you see a different light, you have to peck the horizontal. Green one shows, and then you have to peck the horizontal in order to get food. This is called symbolic delayed matching to sample. These are just some of the examples of investigating short-term memory. One of my good friends when I was at the University of Hawaii was one of the first people to really investigate short-term memory in honeybees. So even insects have the ability to have short-term memory. You might consider this a working memory because the animal has to solve a problem ultimately. So if I see the red light, or the green light, I have to remember, to solve this problem, I have to go to this other one. So there are two main factors that affect how well animals and humans do on delayed matching to sample tasks. The length of the sample, how long it's presented, and the length of the delay. The longer the sample is presented, the better they do, and the longer the retention interval, the worse they do. So this was a very famous study on delayed matching to sample by Grant in 1976. This is how long the sample was presented. 14 in red, 8, 4, and 1 second. And this is the delay. The sample was presented for this amount of time, and then there was no delay until they had to choose from the two others. And you can see without any delay, they all do really well. But the animal that got to look at it for 14 seconds, they got it every time. But an animal that only got to see the sample for 1 second, not as good. But as the delay increased, 20 seconds they had to keep in short-term memory, you can see that all groups dropped. 40 seconds, even worse. 60 seconds, and we're getting almost to chance because we had essentially two things to choose from after the sample is presented. So if you present the sample for 1 second, and then they have to wait 60 seconds to choose between the two objects, one was the sample and one was something new, you can see over here, they're at a little over a chance, 52%. Now, I use delayed matching sample in some of my experiments. And so what I do is I use images, different images, pictures that I've taken that are very similar. Some are very difficult, some are very easy. So another factor that influences delayed matching sample is, of course, the similarity of the sample to the things that they have to choose from. Now, because I'm working with humans, I got to make it a little difficult. So images flashed on the screen, really flashed. That was only 0.25 of a second. And then they have to choose from here which one they saw. Which one is it? Do you know? It was this one right here. So they choose that one, and I can measure short-term memory, and then I can look at things that influence their ability to remember stuff. I affect their success by how long I present the sample. That was really only a quarter of a second, but what if I presented it for a half a second or a minute? They get better. And then that interval, there was a 1 second interval. If I add that interval, increase that interval, they tend to not do so well. Now, some of the things that I've done with it is like an experiment to look at risk taking. So I present an image very quickly, then 4 similar images. Then they have to choose. Then I ask their confidence. How confident were they got it right? And then I ask them to risk or play it safe. Playing it safe means they take a certain amount of points whether they're right or wrong. Risk, they get a high amount of points if they were right and no points if they were wrong. And I've done a lot of work with this and published a few papers. I can also look at some aspects of short-term memory, but then their decision-making is based on their memory, is the things that I'm interested in. So let's look at another thing called retroactive interference. What interferes with their ability? So here are some factors we've looked at that affect delayed matching to sample. How long the sample is presented, how long there's a delay, how similar the sample is to the objects they have to choose from, and whether there is an interference. So here is what's called retroactive interference. I present a stimulus, goes away. I present another stimulus, it goes away, and then the animal has to choose. In this case, because the green was presented, the green square acts as an interference to their ability to remember the sample. Retroactively meaning this affects what came before it. Adding a retroactive interference diminishes their ability to recall because they're not sure what they need to remember, the circle or the square, and then they get to choose. Proactive interference is very similar, except for the thing that is causing the interference comes first, then the sample, then the choice. That's called proactive interference. The green square proactively interferes with the animal's ability to remember the blue circle. So let's look at it again. I have it all out here. So, and this is proactive. The red light comes on, then a blue, then a delay, and then they have to choose between the blue and the yellow. They have to pick the blue in order to get food, but the animal's also, he doesn't know the rules, and so it's remembering the red, and it's remembering the blue, and that's difficult. Proactive interference. It might be like you have to study for an exam, and your exam is in biology. So you study a bunch for a biology exam and then take the biology exam. But if you study, before you study the biology material, you study stuff in psychology or English, then that information can proactively interfere with your memory of what you're going to be tested on. Here's retroactive interference. The sample is presented, then something comes as an interference. That interference retroactively interferes. There's a delay. And again, the animal has to choose the sample if this is delayed matching to sample. Now, interference can tell us a little bit about the serial position effect. Remember the words you had to remember? Most people tend to remember the first ones and the last ones, but the middle are difficult. And one of the ideas of why we get this sort of U-shape distribution of memory is because of what these stimuli are dealing with. They are only dealing with retroactive interference. In other words, people are trying to remember spoon and sink, but nothing really came before them, not a lot anyway. And so when they recall at the end, there's not a lot of interference with these first ones. And also not a lot of retroactive interference with these last ones. Because as soon as apple and can are presented, there's not a lot of things that are causing interference and they remember them. But the ones in the middle have both proactive interference from trying to remember the first ones and retroactive interference from the ones that come towards the end. There are other memory systems that are often investigated by people interested in animal behavior and learning. Song learning by birds is a really specialized way of learning, very interesting to do research. Imprinting, the idea of learning which animal is my mother. We've talked about imprinting a little bit. There seems to be kind of a window of time when animals imprint on mother figures. Migration and orientation learning. I mean, these are really amazing things that animals can do. They can navigate across the globe only by learning a path maybe once. And then there's spatial learning, which we've talked about a little bit. And there's a number of ways of investigating spatial learning that I'd like to talk about. So one thing often used to investigate spatial learning is what's called a Morris water maze. And what happens is you have this tub and a small tiny little pool, like a kid's pool. And the water that is filled in this pool, you add some milk or something to make it so that you can't see through the water, something that makes it opaque. And just under the surface, where the animal can't see, is a little platform. And this little mouse does not want to swim. It would much rather find a place to rest because it's a little scared it's going to drown. And so you put it in this maze somewhere, and it swims around, and finally, right below the surface, it finds somewhere that it can stand. So what we find is when we put the animal back in, the little mouse back in this kind of milky maze, a water maze, it takes less time for the little mouse to get to the platform. It doesn't swim all around. It takes a straighter and less twisty route because it wants to stand on there. And what it's doing is it's using information around in the lab to navigate. Okay, the platform's over here and it's near there. And if you make the outside very distinguished in its stimuli, maybe there's a big black stripe thing here and a big thing filled with circles here, it does even better. In other words, it's using the space around it. Eventually, not very quickly, the mouse just swims right to the platform using those spatial cues. And what we find through research is one of the major things is how the hippocampus is functioning. So a lot of research on how the hippocampus helps in navigation, they use a water maze. Another common thing to look at, short-term memory, but also navigation and spatial memory, is called a radial arm maze. And the little mouse is put in the middle, and there's food in these dishes on the outside. And here's another one where the animal's enclosed. You can see the little mouse there. And what the mouse has to do is it has to go out, eat some food, come back, go out, eat some food. It's a hungry little mouse. And the object is, don't go where you've been. In other words, remember all the places you've been, or remember all the places you need to go, because it's a waste of time if at the beginning you go down here and eat some food and then you come back and then you navigate to the others. Before you go to one that still has food in it, you don't want to go over here. You've already been there. Okay, so a mouse goes out and he gets some food at one end and he comes back to the middle. He goes out to another. Remember, he's very hungry, but he doesn't want to go to a place he's already been. So he has to remember, where have I been? Where do I need to go? But eventually the animal will make a mistake. It'll go to an arm that it's already been to. And the question is, where? When does it make the most mistakes? So this research was done in 1988 by Kesner and Despain. Rats were trained in a 12-arm radial arm maze. Once proficient, they knew where to get the food. They knew about the maze. You know, they're hungry animals. They knew about the maze. They were tested with 15-minute interruptions occurring after visiting a specific number of arms. So they got to visit some arms, then they had a break of 15 minutes where the short-term memory is really going to be tested. Question is, when did they make the most errors? When was it the hardest? Was it after visiting only one arm and then they took a 15-minute break and they came back and they made a mistake and they visited that same arm? Or had they already visited maybe 11 of the arms and there's only one left? You take them out for 15 minutes, you put them back, and then they go to an arm they haven't been to. When do they make the most mistakes? What they found was they made very few errors at the beginning. The most errors were about 8. They had visited 8. And they still had 4 to go. But if they had visited 10, you take them out, you put them back in, they weren't too bad about finding those remaining 2 they needed to go to. In other words, the most errors were about in the middle and not at the end or not at the beginning. So what is that telling us? It's telling us that in this maze, the animal is remembering a couple of things. One, it's remembering, where did I go? Try to keep in my memory where I've been, and I know not to go back there. Or it's trying to remember, where do I need to go? Where do I need to go? And what we know is if you have to remember every place you've been, that's a lot. That's a lot to remember. And if that's all you're doing is remembering where you've been, it would be harder and harder. You'd make the most mistakes at 10, 11, right? Because you're trying to remember all those 10 and 11 places you've been. So the animals kind of switch. In other words, if at the beginning you have to keep in memory all the places you need to go, you'd make a lot of errors here. So when you have to remember about 6 to 8 things, you switch from where was I, where have I already been, to where do I need to go. So this is the difference between what's called prospective and retrospective memory, or coding. Sometimes it's called prospective coding or retrospective coding. Prospective coding is keeping in memory the things you still have to do. Right? Keep in mind where I need to go. This is what we see is the animals shift in the middle to prospective coding because there are fewer things to remember of where I still need to go during that 15-minute break. So if I've already visited 10 arms, it's really easy. I'm not going to make mistakes if I just keep in mind those are the 2 I still need to go to. I don't care about where I've been. Just keep in mind where I need to go. At the beginning, it's easier to keep retrospective coding. Where have I been? I've been to those 2 places. Just keep that in mind. I've been to those 2 arms, and I don't want to go back to those 2 arms. Go anywhere else. I don't care where you go. Don't go to those other 2 because that's a waste of time. You've been there. Again, the animals switch from retrospective coding at the beginning to prospective coding at the end because it's just easier.